Located in National Jiao University, Hsinchu, Taiwan, we meet all our customers' diverse needs through the combined efforts of biotechnology, engineering, and information technology. Apollo is dedicated to immunology. This year, we have a beautiful encounter with IGEM. We integrate bacteria and antibodies into a powerful probe that will redefine detection. Apollo can achieve all that you need, the way you need it, with more precision using less time. We reinvent ourselves every cycle. Every facet of our process is revolutionary. We continue to transform the way people do things. We simplify, we perfect, we start over. We make the difference. The only thing we can detect is everything.
Participating in our product announcements. I'm CEO Jenny. Our company, Apollo, stands for Almighty Probe of SCIV Organization. It also means Archer, which implies our principle. We want to use antibody to do detection in a really precise way. And we have created a platform that will transform any desired plasmid and color signal. There will be infinite possibility. Imagine company, hospital, or people could order their detection probe just meet their need. And imagine we can build millions of probes to satisfy everyone. These are not future technology we've seen in Iron Man. These are something we have right here, right now. And we call it EcoTactor. And we'll also improve antibody production, which requires at least 30 days, and experimental methods needed. More detection costs thousands of dollars. All this problem could be solved by synthetic biology, use E. coli to express recombinant antibody, scFv. And scFv is a fusion protein that's composed of only variable region and linked peptide, but it still retains a function. So we use an easy way to produce a large amount of scFv with little cost. An EcoTactor could apply to various uses. For example, we can apply it to health and medicine, agriculture, food industry, and environment. And we want to focus on the most important topic, which is health and medicine. We found that detection for targeted drug is quite necessary. And how does targeted drug work? It will circulate throughout the body until it can find and hook onto the targeted marker and accurately kill the cancer cell. So targeted drug is more specific and does little harm to human body. And the percentage of using targeted drug in 2003 was 11. After 10 years, it grew to 46, which is 4 times larger and half of the market in cancer treatments, which means pre-diagnosis of targeted drug is demanded. And also we talked to the doctor told us the patient have to undergo detection process before using targeted drug. So the first step will be the doctor diagnose the patient have the cancer, and here comes our EcoTactor to determine the right targeted drug and immediately start the treatment. And FDA also approved combination therapy, which is using multiple targeted drugs at the same time. This will definitely benefit everyone because there will be fewer side effects and lower treatment failure rates and lower case fatality rates. We also do some survey on public recognition of targeted drug. Over 50% of people know the importance of pre-diagnosis of targeted drug. Over 60% of people recognize combination therapy. Over 90% of people believe that pre-diagnosis and combination therapy are worth developing. So this year, we have created a customized platform that could transform any desired plasmid for prescribing combination therapy. And this is a revolution. The only thing we can detect is everything. To get more details, please allow me to introduce you Chief Technology Officer Ray.
Good morning, everyone. I'm Ray. We have mentioned that we want to assist the doctor to guide the patient in using targeted drugs. But how do we make it? We want to use targeted drugs to directly detect whether the patient is suitable for targeted drugs, because this method is more direct. So here comes our EcoTactor. In our product, we use 3 different monoclonal antibody targeted drugs, which are Herceptin, Erbitux, and Avastin. They correspondingly bind to human epidermal growth factor receptor 2, such as HER2, epidermal growth factor receptor, such as EGFR, and vascular endothelial growth factor, such as VEGF. HER2 and EGFR are cell-surface receptors whose overexpression will lead to uncontrollable cell proliferation. However, VEGF is not a cell-surface receptor, but a growth factor, which will lead to angiogenesis and cause cancer cells to metastasize. We use the scFv of these drugs because scFv is smaller than antibody. It won't place any stress on E. coli for expressing it. To display an scFv on the E. coli surface, we use transmembrane protein. The transmembrane protein is composed of lipoprotein, such as LPP, and other membrane protein A, such as AMP. The C-terminal of transmembrane protein is connected with the scFv and bring it out of the E. coli surface. In our product, this LPP-AMP, that makes a difference. We improve the function of existing LPP-AMP, allowing our customer to change the scFv at will. What's more, we built the scFv library. We've constructed 3 different scFv, which are anti-HER2, anti-EGFR, and anti-VEGF. To express the color signal, we took fluorescence protein and chromoproteins from nitrogen kit. And then we put 2 plasmids, which are scFv and color signal in one E. coli by co-transforming. Therefore, our customer can choose whatever combination they want. Besides, our eco-detector has different function and different color signal, can target different markers on the cancer cell simultaneously. By this, the best combination of therapy could be prescribed. Next, I'm going to show you a video about our customization procedure.
Hello, may I help you?
Dr. Lin, I would like to order some products of yours.
Which product would you like to order?
I would like to have 3 probes with different colors, detecting HER2, EGFR, and VEGF.
Thank you for your order. We will ship your order in 3 days. Your certificate of analysis to go with.
Dr. Lin, I heard my orders already.
Yes, we have it right here. We shipped it out just today with the certificate of analysis.
Oh, well, that's great. I really appreciate your hardworking and awesome speed. I definitely will order from you guys next time. Thanks.
Thank you, Doctor. Hi, I'm Product Manager Morris. As you can see, we provide a fast ordering procedure. Next, let me introduce our certificate of analysis. In order to put our product into practical use, we need to kill our ecotector, but also maintain the function of our protein. We chose tetracycline, which is a kind of antibiotics, to meet our goal. In our experiment, we incubated our ecotector with different concentrations of tetracycline. And we found out that 100 micrograms per milliliter of tetracycline can fully kill our ecotector, since there is no bacterial colonies found on the plate. Next, we incubated this certain concentration of tetracycline with our ecotector in LB growth. We found out that untreated ecotector grew rapidly in growth. On the other hand, the treated didn't, and the protein was also maintained. This means with tetracycline, our ecotector is a functional and usable product. Next, let's check the result of our ordering. We built 3 combinations. The first one is anti-EGFR with green fluorescence protein, anti-VEGF with red fluorescence protein. We not only use fluorescence protein as our signal, but we also combine our scFvs with chromoprotein. So in our last combination, we combine anti-HER2 with blue chromoprotein. To prove that our ecotectors can actually bind onto the cancer cells, we did our cell staining experiment. In our experiment, we added our ecotectors directly into the cancer well, and we detect the color signal to analyze the binding ability. In our first cell staining experiment, we used a cancer cell known as SKOV3, which overexpresses EGFR. The blue color is DEPI, which is used for cell nucleus staining, and the green spots are our anti-EGFR ecotectors. Compared with the control, you can see that our anti-EGFR ecotector has successfully bound onto the cancer cells. Next, in our second cell staining experiment, we also use SKOV3, since it also expresses the VEGF receptor. The blue color is also DEPI, and the red spots are our anti-VEGF ecotectors, which are located in the cytoplasm of the cell. This means that our anti-VEGF ecotector has also successfully bound onto the cancer cell. Lastly, in our third cell staining experiment, we used another cancer cell known as SKBR3, which overexpresses the HER2 receptor. And as the image shown above, our anti-HER2 ecotector has also successfully bound onto the cancer cell. This means that all three of our ecotectors are very precise probes, and the doctor can therefore prescribe the corresponding targeted drug. We not only built these three combinations, but we also connected our SCFVs with all three colored signals. And this is a result of our amazing customized detecting platform. To introduce our manufacturing efficiency, I will welcome our R&D Department Manager, Amber.
Hello everyone, I'm R&D Department Manager Amber. Our modeling is to find out the optimum SAV production time. We choose to use the Hill function-based model for our modeling. From this graph, you can see the orange curve is a simulation curve that we used several experiments to find out the optimum SAV production time. And next, we conducted another experiment to validate our simulations. You can see that these two curves rise in a similar way, which means that our modeling is very accurate. And we not only modeled green fluorescent proteins, but we also modeled red fluorescent proteins and blue chromoproteins. And we successfully found out the optimum SAV production time for each color. And now, we have prepared everything for our SAV ecotectors, from designing SAV as probes and adding another color signal for observation, conducting cell staining experiments to guarantee its function and performance, and also considering the safety for our customers and its efficiency of modeling. But we reinvent ourselves every cycle. We not only constructed SAV plasmid, but we also developed another plasmid into our library. This amazing plasmid as building blocks can connect an organism with solid material. It is called gold-binding polypeptide. Gold-binding polypeptide has strongly specific binding affinity to the gold chip. It is more flexible and better immobilized on the gold. And we selected the third repeated sequence of gold-binding polypeptide and links with fat L. Long-chain fatty acid transport protein, which is a kind of transmembrane protein that can bring our gold-binding polypeptide to the outer membrane of our ecotector. And since gold chips are usually used as transducer in biosensor, this opened up more possibilities of our Apollo's ecotector's application. And now, I want to check the result of our gold-binding polypeptide. To prove our GBP ecotector can really fix on a gold chip, we designed our experiment with a tiny gold plating chip. We used GFP E. coli as control and compared it with E. coli expressing both GBP and GFP. Then, we observed the fluorescence on the gold chip to analyze the binding ability. Compared with the control, as you can see, E. coli expressing both GBP and GFP has successfully and massively bound onto the gold chip, which means that we build our parts correctly. So we have built this powerful product, but without promoting it, we can't influence our customers. So now, I want to introduce you to our marketing department manager, Tina.
Hello everyone, I'm marketing manager, Tina. I want to tell you that our company not only focuses on the technical part of our product, we also understand that our product will eventually be used by people. That's why this year, our policy and practices are engaged in educational, social, legal, medical, and economic dimensions. For the educational part, we participated in holding a biocamp for senior high school students in July. And the purpose of biocamp is teach high school students synthetic biology. And also, we promoted the IGEM competition by conveying the essence and the values of IGEM. And for the social part, we held the Asian conference. We had 30 teams from China, Hong Kong, Taiwan, and India, and Kazakhstan. And during the processes, each team presented their project idea, which prepared themselves for the giant jamboree. And what's more, we conducted an interesting activity, Be Bold, to let the people know the core value of our product and the importance of our goal. For the legal part, we consulted the Institute of Technology Law to learn that if one day our product will be patented, what legal problem will we face? And for the medical part, since we provide a new medical method, we consulted an oncologist to learn about currently how hospitals handle the targeted drug, which tell us how we contribute to the society. The last, for the economic part, we went to Apex Bio Company to share our EcoTector and learn how to improve it, since we want to provide a more complete product to our customer. These are what we have done for the policy and practices this year. Now I want to bring the stage back to our beautiful CEO, Janet G.
So that's wonderful. Please allow me to remind you how amazing our product is. Our company, Apollo, this year have created a customized platform by co-transforming any desired plasmid. And we have created 3 EcoTectors displaying 3 SCAV, anti-HER2, anti-EGFR, anti-VEGF. And all of these are from monoclonal antibody targeted drug, which make our EcoTector a more direct way to do detection. With different SCAV probe, we have different corresponding color signal. By observing the color, we can know the marker contribution on cancer cell, and therefore determine the right combination of targeted drug. In addition, we expand library with go-body polypeptide. With GBP, our EcoTector can have more use. And SCAV is changeable, so we can not only detect these 3 markers, we can detect anything we want, simply by changing the SCAV sequence. Moreover, we can co-transform any color signal or any plasmid from the library. We use the technique co-transformation to create almighty probe of SCAV. We also achieved the standard of IGEM. We have submitted 23 biobricks. 7 of them are basic parts, include 3 SCAV, anti-HER2, anti-EGFR, anti-VEGF. And 1 GPP, 1 transmembrane protein, LPP-OMP-A, and LPP-OMP-A-N, and FADL connected with GBP. And we also submitted 16 composite parts composed of 7 basic parts. Some of the biobricks are made improved from the existing biobrick. And all our experiments have shown that SCAV could specifically bind onto the cancer cell. We also held some events like Be Bold to let more people know more about our product, and this company to know more about markets, and talk to the doctor to know more about practical use. We help other registered teams, and we help Mingdao to analyze protein amino acid, and we help HSNU to clone biobrick, and we provide the Morano with our own biobrick. We also help NTHU to form their first IGEM team. They are going to join Giant Jamboree in 2016. At the end, I want to thank you for participating in our product announcement. And I want to thank all the people who work in Apollo and who have worked so hard on making this product we'll sing today. The only thing we can detect is everything. Thank you.